Thank you very much. It's really terrific to be here. I've had a wonderful morning already talking with people. It's been a real pleasure learning about Fiocruz, and I'm really delighted to be here. What I want to talk about today is the story of my involvement with this rather wonderful protein, the green fluorescent protein, or GFP. But I want to start in a somewhat unusual way. I want to start by telling you all the things that I was taught that were wrong about scientists when I was growing up. Now, I wasn't actually given a test on all of this. I was really given was told about and read about all the great scientists of the past. People like Newton and Galileo and Fleming and Darwin and all these wonderful, Einstein, all these wonderful scientists, and their lives and what they did in science gave me an idea about what it meant to be a scientist and how to be a scientist. And I realized a couple of years ago that all of these things that I was taught and believed in were, if you listen to the story of GFP, you find out that every single one of them is wrong. And so let me show you the list of all the things that are wrong, and then I'll tell you the story and you'll see why they're all wrong. Although I suspect when I give you the list, you'll understand that they're wrong from the beginning. So, what I was taught as a scientist, going to elementary school and high school, listening to stories of scientists, the first thing is that scientists are geniuses. They're a special type of person. And they have this innate ability to do science. It's just, they're born with it. So if you're not born with it, forget it. It's no good. This is a little bit like they sometimes say of sports people or musicians, that they have it as an innate ability. They have natural talent. It's natural talent and a lot of work on top of it, really. But scientists are geniuses. So that was one of the things, a fear that I had in my mind, that I didn't quite make it into this class. But anyway, there were other things that I learned. The next thing that I learned is that scientists experiments, whenever they work immediately. So you have an idea and boom, the experiment works and you're in really good shape after that. And maybe the reason that they did that the experiments always work, because that's all you ever heard about, we didn't have long enough times to tell stories where the science didn't work, is that the scientists actually used a very peculiar way of thinking. A way that no one else thinks like, but scientists do this, called the scientific method. You come up with a problem, you think about the problem, you come up with a hypothesis. Once you have the hypothesis in mind, you think of the perfect experiment, you do it, it works the first time, then you get a bunch of awards, or you get written up in the textbooks. So they use this method. This is how science is done. Purposeful. You know what you're going after, and you do the perfect experiment to answer that question. Next thing, and this was about all the scientists, actually it started to break down sometime in the late 1960s, because then high schools and elementary schools started talking about Watson and Crick. But before that, it was clear from all the examples that scientists work alone. They never work with other people, except if you watch a lot of movies, you realize that they actually have an assistant, usually named Igor. But they don't work with other people. And finally, all the examples that I heard of, virtually all the examples, except for Marie Curie, every single one of them was a white man. Someone was European or of European descent. There were no scientists anywhere else in the world. And so, you can tell all of these are false to begin with, but now I'm going to tell you the GFP story anyway to back this up. My involvement with this protein came about because I happened to go one day to a seminar and hear a rather interesting story. But before telling you that story, I want to tell you where I was in terms of my scientific thinking. For years, I had been working on this small nematode called Cenorhabditis elegans. And this animal has one feature among many that make it a useful experimental organism. It has one that's really wonderful. It is transparent. So this millimeter-long worm, you can look down the microscope and see every single nucleus in the animal. So we know all the cells of it. We know a lot of other things about it. We have been interested, as was mentioned before, in the development of the nervous system, and in particular of some cells that were the sensors of touch. I won't go into the reason why we were interested in that, but we were starting to find mutants that were touch insensitive. And as we got the mutants, we then cloned the genes that were mutant. And the first question we wanted to address was, where in the animal were these genes turned on? Where were the genes activated? Were they in the touch sensing cells, or maybe they were in other cells? And we wanted to know how we could see that. And at the time, we could easily do those experiments. There were 3 methods that would allow one to look at gene expression. Because we know DNA makes RNA makes protein, it was easy enough to make an antibody against the protein and ask in what cells that antibody did that antibody stain. Or we could look at the RNA by using in situ hybridization, or we could make reporter constructs in which we took the gene and had it make not its normal product, but something like the beta-galactosidase from the E. coli gene LAC-Z, which Malcolm Casadabon in the early 1960s developed as a wonderful way of looking at gene expression. And so we could do that. It produced, wherever the enzyme was, you get these wonderful blue spots, and you could see immediately what cells generated that turned on the gene. The problem with these experiments, however, is that this required a lot of work. But what I mean by that is we had to prepare the specimens. We had to fix them, which meant they had to be killed. Everything had to be fixed so that nothing moved around. Then we had to be able to get the antibody into the animal, or the probes for the in situ hybridization, or the enzyme substrate for this beta-galactosidase activity. And that meant we had to permeabilize the tissue. And all of this was a lot of work. And because the specimen was dead, it gave you a snapshot in time, a single static point. If we wanted to do this again, we had to repeat the entire procedure all over again. If we wanted to look at it at different times in development, for example, we'd have to prepare samples for every different time in development. So we didn't get a very nice time resolution. But it answered the question. So this is not one of those stories where I had an insurmountable problem and I came up with a brilliant idea of how to solve it. We solved it. We knew the answer. But in April of 1989, I went to a seminar, and at that seminar, I heard about the work of Osamu Shimomura, who shared the prize with me. Before leaving this slide, though, I want to point out something about the color code for people's names. You're going to see a fair number of pictures, and there's going to be a color code. Anyone whose name, as these 2 people, Hong Ping Du, who is a graduate student at the time, and Shohei Matani, who is a postdoc, anyone whose name is in red is someone that worked in my laboratory. You'll see some people whose names are in blue. Those are collaborators. And, as you'll see in this next slide, some people will have their name in black. These are the people that did something I wish I had done. So I'm listening to a seminar by Osamu Shimomura about him and about his work on this jellyfish, Aquaria victoria. If you have a little bit of time, 29 minutes to be exact, please go to the nobelprize.org website and listen to the Nobel acceptance speech that he gave, which is 29 minutes long. His story is remarkable. But I've learned after being in this and talking to him, when he was 16, he was told, you have to quit school. That's it. Quit high school. You have to work in a factory. He had to go and work in a paint factory that was actually outside the city he was living in. It was over the mountains in the adjacent valley. But he was sent there to work. This actually turned out to be good for him because the time was 1945 and the city was Nagasaki, Japan. And by being on the other side of the mountains, he was protected when the atomic bomb destroyed the city. He went in and helped people, but he couldn't go to college because there was no college. Eventually, they started to rebuild the university, and the first place that he could go to was the pharmacy school. He said that he wasn't that interested in the pharmacy school, but it was the only college he could go to. He then got a job as a technician. And for those graduate students out there listening to this, this is the unfortunate, sad part, because this is where I admit that he was given a project that those of us that run labs all too often give to graduate students and technicians. And that is a project that we know has already failed with several people. But we hope that there will be somebody that will come in that will solve it for us. And that's what he was given. But it was a fascinating problem. There's a small crustacean that lives off the coast of Japan called Cyprinina. And Cyprinina is a bioluminescent crustacean. There are lots of organisms that generate light: fireflies, glowworms, bacteria, fungi, fish, crustaceans, jellyfish, and they all seem to generate light in different ways biochemically. So while people knew how fireflies generated light, they had no idea about Cyprinina, and that was his project. And he's a very good biochemist, and he was able, in fact, to solve the problem, and he was able to do this. This led to two different consequences. The first consequence was he was invited to come to the United States to do some more research. And the second consequence was that as a going-away present, his boss at Nagoya University decided to give him, as a going-away present, a PhD. So just a small little token of his esteem. In any case, Shimamura came to the United States and started working at Friday Harbor Lab to try to figure out why this jellyfish, Aquaria victoria, produced light. And he worked really hard at this. And every time he did the experiment, he did the preparation to isolate the protein that was bioluminescent from this, it failed. And it failed, and it failed. All summer it failed. He never got it to work until one day he was probably very tired, frustrated, and probably hungry because he had worked all day. It was already dark outside, and he decides, it's failed again, I gotta go home and get dinner. Takes the prep, and he cleans up after himself. He throws it in the sink. Now, the sink also had probably jellyfish parts in it and some seawater and other things. He throws it away, turns off the light. He's about to leave the room. He looks back on at the sink, and he sees to his amazement that the sink is glowing brightly. I just want to point out that this is a time-honored biochemical scientific method. You throw it on the floor, you throw it in the sink. It's a complete accident. But he thought about what was in the seawater. He said there must be calcium, and that led him to isolate the protein that he names after the jellyfish, aquarin, and he realizes that aquarin plus calcium will give light. And so he's very happy about that. And in fact, aquarin became the first biological calcium indicator. People used it really all over the world. But when he looked at it, his joy was sort of short-lived. And the reason it was short-lived is because the light was the wrong color. The jellyfish produced a green light. This protein produced a blue light. There had to be something else. And he reasoned that there wasn't going to be another protein that generated light, was generating green light, but maybe there was a protein that converted the blue light, or the energy needed to make blue light, into green light. In other words, a fluorescent protein, one that would absorb light of one color and give off light of another color. So taking a handheld ultraviolet lamp, he looked at all of his protein fractions and found that there was another one that had the property that when he shone light on it, it gave back green light. He wrote about this in his 1962 paper announcing the biochemical isolation of a quarin in a footnote, footnote number 3. And he said, I found this other protein. I call it the green protein. We, of course, now call it the green fluorescent protein. And with GFP, green fluorescent protein, a quarin and calcium, now green light is produced. So another accidental discovery. No one even imagined that there was a fluorescent protein. And I'm listening to a seminar about this. And remember, I want to know where genes are expressed, and I work on a transparent animal. I had probably given 10 years of seminars saying, I work on a transparent animal. So finally, I even believed it. And I realized that if that was so, I could put this protein into the animal, and anywhere this protein was made, I could shine blue light onto it, and I would see green coming out. In other words, that it would serve as a lantern to allow me to see things. But it wouldn't have to be in an animal that was fixed. It could be in a living animal. And I started imagining all the experiments I would do if I had such a tool. I was very excited, so much so I do not remember another word of the seminar. I just ignored the whole rest of it and just fantasized. The next day, I was able to get in touch. I did what most biologists would do. I got on the phone. And I called around to find out who was in the process of isolating the DNA for GFP. And I found that that person was Douglas Prasher. And I was very excited. And Doug and I talked for about an hour about what we would do when he finally got the DNA, which he didn't quite have at that point in 1989. We lost track of each other, but 3 years later, a new graduate student, Gia Skirkin, came into my lab, and she had just finished a master's degree in chemical engineering working on fluorescence. So the perfect person for the experiment. I told her about the experiment, this crazy idea I had about putting fluorescent proteins into the worm and seeing where they were expressed. And I said, unfortunately, Douglas Prasher has not gotten in touch with me. I think he failed at getting the cDNA. So we're going to have to look for something else. Let's look online. The university just had added Medline to our computers. We could look things up. And I looked up fluorescent protein, and the first thing that came up was Douglas Prasher's paper saying that he had gotten the cDNA. So I was very excited. We ran down to the library. We got his article. And the article had something that most scientific articles do not have that was absolutely crucial for our subsequent experiments. They had his phone number. So I called him up. We renewed the collaboration, and he sent us the DNA that we could use. And 1 month after Gia had started graduate school, she had this page in her lab notebook, which I've circled the part where she has made strongly fluorescing E. coli. And in fact, this is a picture here from that very first day, from the very first prep. She was able to show that GFP worked on its own in the bacteria. There's another aspect of this slide I like to point out, or this page from her lab notebook. And this is this note up here that says that she used the microscope in the engineering building, which was right next to ours. This is where she had gotten her master's degree. And the reason for this is our fluorescence microscope was a piece of junk. And she tried it, and she wasn't really sure the experiment worked, but she knew where there was a much better microscope. So she took the samples, she went there, and that's where she took this picture, and she was able to see that it clearly had worked. But you see that we had a problem, because if you don't have a microscope and your entire experiments are dependent on a microscope, you're sort of stopped. So I solved this. First, we went to every other microscope in the building that we could use, but eventually we got kicked out of these various labs. And I finally called up all the sales representatives of various microscope companies and asked them, I told them we have just developed a new method using fluorescence of looking at gene expression. We're very excited about it. We definitely need a new microscope, and we're going to buy one, but we don't know which one to buy. And so if they could bring a loaner by the lab and we could try it out, we'd decide whether that was the microscope we were going to buy. And if they could leave it for at least a month or two, we'd give it a really good workout. So we did everything on borrowed microscopes, and then eventually, after the paper was out, we actually did get another microscope. But everything was done in borrowed. Let me tell you, though, how unusual. This is an example of the experiment that was done the first time working. That turned out to be exceptionally important. So it's not exactly the case as with Shimomura, but in this case, it was critical. And let me explain why. Everybody at this time that had worked on GFP, it may have been 10 people in the world doing this, had known that the GFP, the protein, did not need any other extra molecules, any cofactors. It was just the polypeptide chain. And so that's good. That was good news. The problem was that that polypeptide chain did something that most polypeptides never do. And specifically what that was, that a very specific region between serine 65 and glycine 67, these 5 atoms of the backbone cyclized to make this 5-membered ring. And without that, you don't get fluorescence. So this has to be done. And no one had any idea how this rearrangement took place. Everybody thought that there had to be a converting enzyme, another molecule that allowed this change to be made. It might have been 1 converting enzyme, maybe 5 converting enzymes. No one knew anything about the biochemistry. But they were absolutely sure GFP would not work on its own. Now, it did work on its own. We put it into E. coli, there was no problem. It made a fluorescent product. So therefore, GFP is its own converting enzyme. It does the conversion on its own. But if the experiment had not worked, we would have said, they're right. It takes a converting enzyme. I've subsequently found out that 3 other labs were trying to do the very same experiment. And all 3 of the labs, when they did the experiment, it did not work. And so they would have had the wrong conclusion. Let me explain what we did differently from them. So basically, you can ignore all the top part of this slide. One day I'll change it, and just look at the bottom. This is a representation of the DNA that we got from Douglas Prasher, as did these other 3 groups. The green part is the coding sequence for GFP. The red parts are extra jellyfish sequence that happened to be there because he originally cloned a restriction fragment from this EcoR1 site to this EcoR1 site. The 3 groups where it did not succeed wanted to be careful. I didn't want to be careful. I wanted to be sure. And by careful, they didn't want any mistakes in the sequence. The way you do that is you grow up lots of the virus, the phage, and then cut out the DNA at the EcoR1 sites. Now you're sure it's good DNA. And then they went on to test that. But as we now know, there's something in one of these regions that makes it impossible to work. The way we did our experiments, and this was again in 1992, we used polymerase chain reaction. And I said, only amplify the green part, because I just wasn't sure about anything outside of it. And also remember, we were putting it in bacteria. We weren't putting it into one bacterium, we were putting it into millions of bacteria. So if half the sequence, 3/4 of the sequence had mistakes in it, we didn't care. Others would have the right sequence in it, and we'd be able to use them. That turns out to be the critical difference between these various experiments. So they concluded, oh, there must be something else. No one says, oh, it's my fault. And we concluded, well, we're done. It worked. So we were very happy with this. We then put it into worms and then decided to publish the work. And when we did, we sent it to Science. And the reason we sent it to Science is because we didn't want to send it to a specific journal that dealt with cell biology or developmental biology or genetics. We wanted it to go to a general journal that everyone would be able to see. And so we sent this to Science. And that's when the trouble began. Because we sent it, and those of you that have any dealings with this publication know that the editorial board stands guard against any bad papers coming in their opinion. And so the editorial board has to decide whether a paper is worthy to be reviewed. And if they don't think it's worthy to be reviewed, that's it. Because you have to send it somewhere else. And they called me up and they said, well, you know, we're sort of interested in your paper, but we're not sending it out for review. And the reason we're not sending it out for review is because of the title. And I said, what's wrong with the title? And I said, well, you see, your title, green fluorescent protein, a new marker for gene expression, you know, it's nice and short. But the problem is it has the word new in it. And everything that we publish in Science magazine is new. And so I asked them, if I change the title, will you send it out for review? And I think they may have said maybe. I'm not really sure. But I did change the title. And I must admit that I am a person who does not really like to be told what to do. And so the title I submitted was a bit extensive. The Aquaria Victoria green fluorescent protein needs no exogenously added component to produce a fluorescent product in prokaryotic and eukaryotic cells. This is more or less the entire paper. I could have just appended the figures and I would have been all right here. But the reviewers liked it. It got published. And then the copy editor called me up and said, do you know that your title is too long, or it's quite long? And I said, yeah, I could change it. How about this? Green fluorescent protein as a marker for gene expression. And they were very appreciative and grateful that I changed it. The second problem we had with publication was I had sent this picture in, and it's maybe a little hard for you to see this picture, but this is of a living, newly hatched worm. And we had put GFP in the nerve cells we were interested in. And what I really want to point out to you is this nerve cell, that's its cell body process, and that's its growth cone. And I wanted this to show people that you could watch biology happen. You could see in a living organism the growth of nerve cells or anything else you were interested in. And so I was very excited about the picture, and the cover editor called me up and said, we really like your picture, and we'd like to use it on the cover. However, there is one color we really don't like to ever use on the cover because it doesn't come out very well, and that's green. Can we change the color? And I said, I'm sorry. You have to have it green. You just can't do that. And actually, this is a somewhat enhanced cover, a picture of the cover, because if you look at it, it really does look sort of washed out. So they were sort of right, but I just couldn't do that. The third problem arose because we had already told people about this way before publication. It was published in February. We started telling people and sending out samples in October of the year before. And so people were already getting back to me and saying, you know, it works in our system too. It works in dictyostelium, it works in fruit flies, it works in yeast. And that was really nice, and I wanted to add that to the paper. So I asked them, can I include this in our paper? And they said, well, you gave us GFP well before your own publication. Of course you can do that. You can cite that we have this. And they did, all except one person who made a series of excessive demands. She's shown here. The demands were that I make coffee every Saturday morning for the next 2 months, ready at 8:30 a.m., prepare a special French dinner at a time of my choosing, and empty the garbage nightly for the next month. This is my wife. But what Tula did was the next really wonderful experiment with GFP. So even though she claims I have not actually ever paid up on this, it was really quite wonderful. Because what she was interested in was a protein that was made in Drosophila. Now, when the Drosophila oocyte is being prepared, there are, so here's the picture of the oocyte, and it grows out and is added to by products made by 15 other cells called nurse cells. They produce proteins that then are brought into the cell. And in the case of the protein she was interested in, as you can see some of it coming in here through what's called a ring canal, the protein comes to lie along this surface of the oocyte and along this surface. And she could watch this protein. So she made the first protein fusion. She added GFP to the protein, and wherever the protein went, she could see it with GFP. And it was a spectacular result, and she published her paper about 4 months after our paper came out. So it was quite wonderful. I will tell you that some of you may come out of this talk, and this is the only thing you're going to remember. I know this because I gave a talk similar to this in India a couple of years ago, and there was a reporter, and this is the only thing he wrote about for the article. We have a local pharmacy that we use, and I went in there some months after the Nobel Prize, and one of the pharmacists came up to me and said, "Are you the guy that won the Nobel Prize?" And I said, "Yes." He said, "Congratulations." I said, "Thank you." And then a couple of months later, my wife went in to pick something up, and he said to her, "Excuse me, aren't you the wife of the guy that won the Nobel Prize?" And she said, "Yes." And he said, "Well, I just want to tell you, I loved your letter." In any case, why was GFP such an exciting thing to work with? Well, I'll give you 4 reasons here. The first is that GFP is encoded in DNA. So if you put the DNA for GFP into an organism, it and all of its subsequent progeny are going to have the GFP. You do one prep and it's there. Second, shining blue light is fairly innocuous. It's basically a non-invasive way of examining an animal. And so shining blue light doesn't change its biology too much. And so we get a chance to really look at the biology. Third, it's a relatively small molecule. In comparison, the beta-galactosidase, which is an obligate tetramer, GFP is a monomer, and the difference in molecular weight between these functional molecules is 16-fold. As a result, beta-galactosidase never gets out of the cell body, whereas GFP can go everywhere, diffuse everywhere in a cell, and outline the entire cell. And finally, as I've said several times here, we can look in living cells and organisms and finally get a dynamic view of the problems we're interested in, rather than the static view that we had before. To give you some examples of organisms with GFP, this is the entire nervous system of the worm we work on, Ceterabditis elegans. These are canola plants, fruit flies, mice, zebrafish. This is Alba, the GFP bunny. Alba was commissioned from a French biotech company by the Brazilian artist Eduardo Koch, who used to take Alba, who was a family pet, to his various art installations to get people to talk about the connection between art and science and art and technology. And unfortunately, Alba lived a nice long life, but is no longer with us. I've heard a rumor, but I don't know that it's true, that he has a green fluorescent puppy, but I don't know. On the right-hand here side, we have several cells. I just want to point out this one, the Purkinje cell back in the cerebellum of a mouse. And you can see how GFP has outlined the wonderful dendritic arborization, the cell body, the axon coming out. Just an astonishing way to look at cells by completely labeling them, because GFP can go everywhere. I've said several times now that GFP is important by being able to look in living organisms. I want to give you 2 examples that were both put on the American Society for Cell Biology website, which I took from there. They're done by a woman named Rosalind Silverman Gavrilla. My understanding is that she made these when she was a graduate student in Canada. And what she's done in these movies is to take GFP and put it on various other components. The first one she did, which is going to be over here on the left, is she took GFP and attached it to a component of the mitotic spindle. Now, the spindle only appears when nuclei divide. So the spindle only will appear in this short period when the division is taking place. The other thing that she did was take GFP and add to it a small polypeptide called the nuclear localization signal. Whenever this signal is on any protein, that protein gets sucked up into the nucleus. But the nucleus is only around before and after the cell division. So during cell division, this protein is going to go everywhere in the embryo. And she looked at this in the early development of Drosophila. And at this time, there are no cell boundaries. There's just nuclear division. So let's look at the left-hand side here. And it's very speeded up to get many divisions here. But you saw the spindle form there. It's going to reform again, another round of division. And it's going to go again. And if we're lucky, it's going to go one more time. And what I hope you see, well, I hope you understand that she named this movie In Synchrony, because all of these divisions are taking place at the same time. And it immediately introduces the question, how does this synchrony come about? What allows this all to happen? And so being able to see something speeded up like this or in real time, a dynamic view, opens up questions that you never even imagined were there. Now, the second movie that I'm going to show from her is a movie that has been falsely colored. And the convention for false coloring is that the more you have, the further along the rainbow you are. So if you have a little, you're blue. If you have a little more, you have green, and then through yellow, and then orange and red is the most. And she calls this movie, after the Van Gogh painting, Starry Night, and I think you'll see why in a moment. So here we go. Nuclei break down for cell division. Now it's all over the cell. The nuclei are going to reform, sucking up the GFP, and then they're going to break down again. And here the pattern is a little bit different. It's not synchronous. It's actually going as a wave that starts in this direction and goes up in that direction. We actually believe that what this is a problem with the prep that she did. That actually this should also have been synchronous, but the cover slip was a little heavy on one side or another, and as a result, this mistake, it's a beautiful movie, but biologically it's not there. But what this says is that GFP could allow someone to actually see how good their prep was, how true to life it really was, and not altered by the artifact of having the cover slip too heavy on it. In my own lab, we've used GFP in a number of different ways. I hope. We've used it to look where genes are expressed, and so many of the genes that we study are genes that are needed for touch sensitivity. And here's one example of a gene that is expressed only in the touch sensing cells. Here's another gene that's needed for touch sensitivity, and it's not in those cells at all. It's in the muscle cells. Completely different cells that have to interact with the touch sensing cells. We can also look, as my wife did, at where proteins are located in the cell. And we can see that this protein, which happens to be the sensor for touch, is actually not evenly distributed, but seems to be more punctate in its distribution along the cells. But once you label cells, you can do all sorts of things with it. We've used it to identify cells for electrophysiology. We've been able to break open the animal, culture the cells, and only the touch sensing cells in culture are the ones that have GFP in them. And then we can put them through a cell sorter and isolate the cells and then study those cells. And we've been able to do that. But one of the things that has been very useful for my lab has been to take this animal, because you saw that picture before of the transparent animal, right? It was just basically all gray. You couldn't really, unless you were very careful, see the cell, and you certainly couldn't see the whole outline of the cell. But now with GFP in these cells, we see these cells very clearly. And once you can see something, you can study it. So we take these animals and we treat them with a mutagen, and we look for animals that have defects. More cells, less cells, cells in the wrong place, cells that have incorrect processes, and so on. All of these are basic problems of neuronal development that we can now address simply because we can see the cells. And so it's been extremely useful. This is one example here at the bottom. Here's a cell that normally has a single process. There's a second process behind it. This one, however, actually splits. You see right here the process coming out here, and it actually splits into two processes, something we never see normally. So again, we now have something to really study that we were not able to obtain before because we couldn't see it. So there's a wonderful quote from the American baseball player Yogi Berra: "You observe a lot by watching," and that really is true here. GFP is wonderful. I consider it one of the most wonderful molecules in the universe, but you know, scientists are never satisfied. They always want more, and the person who shared the prize with us, who gave people more, was Roger Chen, who, among other things, developed fret-based screens to be able to make molecular monitors that would look at calcium, the amount of calcium in the cell, and other tools, enzyme reactions, and so on. And this was all based on the fact that he wanted to produce a lot of different colors, which he did. This is a subset of the colors he produced. And he got tired of naming them after letters. He didn't want it to be YFP for yellow fluorescent protein or GFP for green fluorescent protein. He wanted to name it after something else. So he named all his things after fruits, which he thought would be better. And I think this is, I think that's blueberry, melon, lime, lemon, or banana, I'm not sure. Orange, and that's either tomato or cherry. So all these different, there's about 15 different colors. Now, if you have a lot of colors, of course, that means you can look at a lot of different things. And Josh Saines and Jeff Lichtman at Harvard realized we don't need to use all these different colors. We just need to use 4 that span the whole spectrum. And if we express them in a way so every cell gets a different proportion of the 4, every cell will have a different color. And if we do that, we can label all the nerve cells in the mouse central nervous system. So if you're going to use all the colors of the spectrum and you're going to label all the cells in the mouse nervous system, there's actually only one word you can use to describe this method, and that's brainbow. And these are some of their pictures, and I think you have to admit, these are rather spectacular pictures. Ways of looking at neuronal connectivity and the arrangements of brain segments and so on. And it's really become a very important tool, not only in studying mouse brain, but also zebrafish and other organisms as well. Now, GFP has been used by lots and lots of people. I had the good fortune of talking with some of the people from Chemistry Abstract last year, and I said to them, you know, I don't have any idea how many people have actually used GFP in the 20 years, well now 20 years, since we published our paper. And that's because if you go to Medline, PubMed, you look at the database, people don't use GFP in their titles anymore, or in their abstracts, or in the keywords. They just use it in the materials and methods. And so the person I talked to said, oh, well, we index every single word in every article. We can tell you how many articles have used GFP and other fluorescent proteins. And the number that came back was staggering to me. It's in the order, last year, of 160,000 scientific papers have used GFP. Now, there's probably some reviews in there too, but it's still a pretty astonishing number. And so GFP has been used for what I would call fundamental or basic research, where it's been used to look, as we did, with gene expression or protein localization, or as the basis of mutageneses, to label tissues or cells and to isolate them, to look, as I said, in brain circuitry, and to look at interactions between organisms biologically, to be able to label one organism and look at another. But they've also been used in an area that I would call applied, which is not only in medicine to study problems such as metastasis or transmission of HIV and other viruses, other bacterial infections, other inherited diseases where one has a model system for this. But it's also been used in biotechnology. It's been used in drug discovery. We heard today at lunch about, or before lunch, about it being used here for ways of monitoring things. And it's been used in biosensors. And I'll give you an example of a biosensor that I'm quite struck by. And finally, there are what I would call unexpected consequences of the research. People have gone off and found new fluorescent proteins. There have been the Lukyanovs and Mikhail Matz were really avid scuba divers. They loved to go scuba diving. These were people in Moscow. And they decided they knew something. What they knew is that corals were fluorescent. And so they said, I wonder if there's a protein like GFP in the coral. And so they went off, and they were the ones that found the first red fluorescent protein, just because they knew about the biology. But then people have gone off and found fluorescent molecules in a vast number of organisms where they were not expected. There have been other systems. Karl Dieseroff, who's done wonderful work on what these various rhodopsins needed for optogenetics, told me once that the inspiration for him going off and finding these molecules was actually from GFP, that there were useful molecules in other organisms, and he had an idea he wanted to follow up, and he heard about channel rhodopsin first, and everything went from there. There's been new chemistry as people have looked at how this protein actually becomes fluorescent. How does that ring get produced? There's been things in industry that I would never have predicted. Soon after the Nobel, I was asked to talk to the people on the board of the marine biology lab at Woods Hole. And after I gave my talk, Jim Sharp, the president of Zeiss Microscopes, gets up to give me a little, thank you. And he says, you don't know this, Marty, but in 1994, before your paper came out, Zeiss Microscopes was seriously considering stopping the production of fluorescent microscopes. We didn't think they were that important. But after your paper came out, everyone wanted to use a fluorescence microscope. And so you saved that part of our business. And I said, so that means you're going to give me a microscope? And he said, no, here's a binocular. But it was really interesting. And I'll show you an example of GFP coming up in art. But I want to talk first, one question from medicine. So in most textbooks, especially since we talk about bacterial phages, we talk about viruses, we talk about cells being infected by the viral particle, and then that particle being released when the cell lyses and then going and infecting other cells. But if you had an antibody against that, you could scarf it up and you could be able to control this. However, when people started looking at HIV, so here is HIV labeled in a mouse cell, here's another mouse cell that's not labeled, and there's a particle that will eventually be released not to the outside, but actually from inside one cell to inside another. That makes it sort of impossible for an antibody to get at it. And so it means you have to start looking at other things, how these cells are associated with each other, to looking at control. Now, there is apparently some release with HIV, so I don't want to say it's exclusive, but this ability to watch things happen can change the way you address a problem, both in basic biology, but also in health-related issues. I talked about biosensors. There's a man named Bob Burledge. He's now at the University of Wisconsin in Milwaukee, but he used to be at Oak Ridge National Laboratory. And Bob Burledge had this rather interesting idea. He knew that there were some genes in bacteria that were activated by the explosive TNT, probably because it looked like some plant protein that activated them. So he said, I'm going to make E. coli sensitive to TNT, and I'll put the appropriate promoter next to the coding sequence of GFP, and so whenever TNT is around where the bacteria will glow green. And he made that, and it worked in the Petri dish just great. And then he had a friend, because he was at the national lab and could do this, they got 5 landmines. Landmines leak TNT. And this is the whole reason he wanted to do this. He had his friend bury the landmines in a 3-meter by 5-meter plot of land, and he's not stupid. They were not connected. But they did have TNT in them, and they leaked the TNT, and he sprayed the bacteria on that plot of land, and then went back at night with a UV lamp to see that he, in fact, could find where the landmines were buried. This would be a wonderful thing if it worked. There's a lot of problems with it. At least 3 groups I know of have successfully gotten it sort of to this stage, but it cross-reacts with things, so it's not 100%. People are still working on it. But what a wonderful way to take this idea and then apply it to something that can really help people by getting rid of this scourge, this thing that's just horrible about warfare. So, has it been used for other things? Yes, there's a group in Japan that hooked GFP onto fibrin for silk and made fluorescent silk from the silk moths and fluorescent clothing. When I met them, they gave me a GFP tie, but I didn't bring it this time. And finally, I said art. I don't know if you would call this art, but one day the people in my lab ran into my office and said, "You've got to go watch this movie. This is fantastic. You're going to love this." And this is the reported first human transgenic with GFP. And if you watch the movie, there's this jellyfish that goes bobbing along at the beginning of the movie, and a hypodermic comes in, sucks out the GFP, and that's the reason why Bruce Banner, whenever he gets angry, turns on the gene for anger, and GFP shows up in his skin. And I happen to know the screenwriter for this. It's a guy named James Shamus. He works at Columbia, and I talked to him, and I said, "James, I saw the movie. It's terrific. How did you know about my work?" And he said, "I have no idea what you do." And he said, and I explained, and he said, "Oh, no, that. We had a student from MIT working on the set, and he convinced us to do this. I will take it any way I can." Let me summarize by telling you some of the things that I believe. I think I've convinced you that GFP sort of puts a lie to most of the ideas I learned as a youngster. Let me tell you what I believe it has taught me. The first is that scientific success comes in many different ways. I've told you Shimomura's story. Let me tell you briefly Roger Chen's story. Roger Chen, we have this national contest every year for high school students. It used to be called the Westinghouse Talent Search, Science Talent Search, and now it's called the Intel Science Talent Search. Only the best of the best even get to be competing in this. He won it as a high school student. Unbelievably accomplished scientist, was that all the way through college and has continued for his whole life to produce wonderful results. I, on the other hand, will not show you my chemistry grades or any of my college grades, at least not now. Let me just tell you, they were not stellar. They were not something that you would say, "Oh, this guy should become a scientist." Nonetheless, I still liked it. And I think what unites us, all 3 of us, is the real passion that we have in doing the experiments and the excitement that we get from doing the science. The third thing is I think it's, got 2 there, but I'll do one after the other. The first is that many, if not most, discoveries are accidental. And I like to quote Enrico Fermi about this, because Enrico Fermi has this wonderful line that goes something on the order, if you do an experiment and it confirms your hypothesis, then you've made a measurement. But if you do the experiment and it doesn't confirm your hypothesis, then you've made a discovery. Now, of course, you got to do a lot of experiments to get these strange results that then maybe lead you in new directions. But I think it really is true. And if you go and you read the various biographies and lectures by the Nobel laureates, I think you will be somewhat surprised how many of them use the word accident or lucky in the description of the work. They were working on one project, suddenly something appeared, and that led them in a whole new direction. And you never can know when this is going to come up. The third thing here is that I believe that being ignorant, stubborn, and having a willingness to try things is exceptionally important. We barely knew about the fact that our experiments were not supposed to work. It didn't stop us. We were going to try the experiment anyway, and it worked because what people thought turned out not to be correct. Now, that doesn't mean you have to repeat everybody's experiment from the beginning of time, but it does mean that you should be skeptical, and if you have an idea, try it. A number of us have talked about something that I call the weekend experiment. This is an experiment you do when no one else is in the lab, so Friday evening, Saturday morning, Sunday morning, sometime when no one is around on the weekend. And you're trying an experiment that it just is driving you crazy. You've got to try this. It's going to be fun. And if it works, you get so excited, you tell everyone on Monday morning. You just crow about it. If it doesn't work, you just ask them how their weekend went. You let it go. But I think it's really important to give it a try. The next thing I want to point out is that scientific progress is not made by great scientists. Scientific progress is a cumulative process that everyone contributes. I've already named 6 people in this talk that have done amazing work that have made GFP important. Unfortunately, the Nobel Foundation, the Nobel Awards, can only honor 3 people. So we're really representative. But even the 6 is too small a number. It's the hundreds and thousands of people that have used GFP in novel and original ways that have made it more important and given it its importance through all this. And that's what's so important. I often think about GFP as a good metaphor for how we do science. Because just as GFP absorbs light of one color and modifies it and gives off light of another color, we as scientists take in the observations and results of other people, we modify them by our own experiments and thoughts and ideas, and then we give them off to somebody else to do the same thing all over again. And so we're constantly handing off information and modifying it, and it's the cumulative effect that really adds to the importance. The story of GFP, especially the beginning with the jellyfish Aquaria Victoria, and I would say also when we were doing it, since most people still didn't like the nematode that much, using C. elegans, points out, I think, a very important lesson, and that lesson is all life should be studied. And especially here in Brazil with this mammoth amount of biodiversity, there's a lot to be learned. And I think we do ourselves a disservice by looking only at a few model organisms, as if these will be the ones that will tell us the answers, when in fact we're going to get answers from many different ways. And we know this from all the pharmaceuticals that people have found and have discovered over the years. So all life should be studied. And my final point is that, and maybe this is a little heretical here, but let me explain it, is that basic research is really essential. I am not saying that I don't like translational research. I actually do, especially the older I get, the more I really want translational research. However, I think it's essential that we continue the pipeline of information that starts with basic research, because in many cases we never know where the breakthrough is going to be that will give us the insight into a disease or some health issue, and we need the information, and we need more and more of that. So I'm not against translational research, and I'm not just to have everything as basic research. I want a balance to be produced in this. But I want to leave you with my favorite quote about basic research. And this is a quote that was said by Robert R. Wilson, who was a physicist, an architect, and a sculptor, and was the first head and actually the designer of the Fermi National Lab in Batavia, Illinois, that before the CERN Super Collider was the world's largest particle accelerator. One example of how he was as an architect, he wanted to make a tower that would allow people to see the entire area. So he hired a helicopter, and he had the pilot bring him up and up and up. And when he finally got the view he liked, he said, "What's the altimeter? What's the height here?" And the pilot told him, and that's how high they built the building there. So a very original man. But in 1969, and you have to remember, 1969 in the United States, we're in the middle of the Vietnam War. There's a lot of concern about national security and defense. And now there was this bill for the Congress to pay for this enormous science project that was going to be a super collider. And there were some senators that were for it. And there was a joint Senate and House a committee to look at science, and they invited Wilson to come and talk about the importance of this. And one of the senators says to him, Dr. Wilson, please tell us the importance of this research for the national security. How many ways will this help national security? And Wilson looked at him and said, none. Not the answer the senator wanted. So he asked it again. No, nothing. And then finally he said, no, really, in what respect will this help the national defense? At which point Wilson said the following. He said, it is only to do with the respect with which we regard one another, the dignity of men, our love of culture. It has to do with whether we are good painters, good sculptors, great poets. I mean all the things we really venerate in our country and are patriotic about. It has nothing to do directly with defending the country, except to make it worth defending. Thank you very much.
Thank you, Marty, for an absolutely wonderful lecture. I don't know if this is on. Happily, we have time for some questions and answers. So, may I invite any questions from the audience? We have 10, maybe we can squeeze 15 minutes in. So, who has a question? I think we have, do we have microphones to? Yeah, great. Who has a question? I'm going to start while people are thinking. You mentioned that 6 months before you published the science paper, you started giving away GFP to anybody who wanted it. That seems like a very generous thing to do. You were not worried that you might be scooped by one of these people?
No, I mean, we had already, on October 1st, we have a newsletter for all the people that work on worms. And I actually said in the newsletter, we've got this thing. Here is the result. We think it would be useful for everyone to work on this. And people from that newsletter article, which is not a citable thing, but it was just something we did, they were able to find out about GFP and they started writing me. Okay. And then they told their friends, and then those told other friends. So we had done it. Now, there was some question at first of, we had developed this new technique of looking at cells and looking at organisms, and we imagined that there might actually be an industrial use of this. And so we did patent this, and you can't patent anything if you make it publicly available. So we very quickly got the patent that would, if any company wanted to use this, then they'd have to get a license. But we weren't going to license or charge people to use it in their laboratories for basic research. So we just started giving it out, and eventually we wound up giving out something like 1,500 samples. And then we sort of got tired of sending out all this stuff, and we just gave it to other people to send out. But no, we wanted people to use it a lot, and we wanted them to use it as soon as possible.
It's very nice that such openness works. I'm not sure if this is on. It is, okay.
Yeah.
Questions, please.
First of all, thank you for your lecture. I appreciate it was awesome. Thank you. I have a question. The philosopher Thomas Kuhn, it's an American one, I think you know him. He said once that there's two kinds of science, the ordinary one and the extraordinary one. And what do you think? The GFP, it's a kind of ordinary science or extraordinary science?
I would say ordinary. There is a question that I have had ever since we started the GFP work. I told you that I heard about GFP at a seminar in 1989. We didn't publish our work on GFP until February 1994. So almost 5 years. It's actually 4 years and 10 months between the time of the seminar and the time that the paper came out. And nobody else in my department, and even more than that, GFP was discovered in 1962. So the question is, why didn't other people have that idea? Partly, I think, is you need to have the molecular biology revolution, and 62 was much too early for this to happen. And then probably people forgot about it. It was an obscure field, bioluminescent. There were few people working on it. Many people probably didn't hear about it. I think that was unfortunate. And but they knew about aquarin. So part of it may have been it took a while for the molecular revolution to give us the tools to be able to do these experiments. The second thing, as I tried to point out in my talk, is that I work on a transparent animal. So if somebody tells you that you can turn on a light and see something inside the animal, if you work on mouse or frog, this is not going to excite you. And I don't know why the other people at that seminar didn't get as excited as I did, but they didn't. And I'm sure that the speaker, Paul Brem, gave seminars at other places. And apparently nobody else got excited at those places either. I think people started to get excited once in 1992. Douglas Prasher had published his paper saying, here's the complete cDNA sequence. And that's when these other three groups started to do their work. But I think people said, oh, well, that's nice, it's fluorescent, and because they didn't work on something transparent, they sort of ignored it. So to me, it wasn't I was saying, gee, you know, I need a way to look at living tissue and looking at things in living tissue. I know, let's get a fluorescent protein and find out if there is such a. I didn't go through that sort of thinking. I was sitting there, too early to fall asleep in the seminar, and all of a sudden somebody says, you can turn on a light in the cell, and it just was obvious. So I don't think I would call it extraordinary. It's not where I pulled together 15 different ideas and synthesized something differently, but I don't know how other people get ideas either. I mean, usually this is the way I get ideas. I have problems that are milling around in my head or things that I've been thinking about, and then suddenly I'll read something or something I have and say, oh, you know, maybe we can do this. So I'm not so sure that's extraordinary science. I think that's what scientists do regularly. So I would say it's ordinary.
Another question.
Hi, I have a more specific question that just came out from my mind. You, in one part of your talk, you're saying that, okay, we are now able to look without interfering because it's just light. But it happens that light modifies biology with clock genes and the circadian cycle. Is there anyone doing research on expressing fluorescent proteins and see if this modifies the cell cycle?
You know, I'm not sure, but you're asking, I'm going to make it a little more general, although your specific question I can't really answer because I don't know if people have done questions like that. But there is a question. If you put GFP onto a protein, as my wife did, and you want to know where the protein goes, you can see where the protein goes. How do you know that's correct? Right? That's a part of this whole problem. How is that correct? And my wife did, in her paper, she did a really wonderful experiment. Because the normal gene that made the protein she was interested in, when that's mutated, fruit flies die. And so she put her GFP protein into animals that didn't have a normal gene. And now they lived. So that said that the GFP protein was able to do everything that the normal protein was able to do. And genetically, we call this rescuing the phenotype, the wild type phenotype. And so she was able to say, what I'm seeing is consistent with the right biology because it has rescued the biology. And so this is a very good way of having more assurance that what you're looking at is actually correct because it replaces something that it has the wild type function.
Hello. I wanted to know what made you be sure that you wanted to be a scientist. What inspired you?
I'll try to make this very brief. When I was in college, I worked in a laboratory and for the whole summer of my junior year. And it starts out the same way as the Shimomura story starts. I kept doing the experiment. It kept failing. In his case, he threw it in the sink. I didn't have that intuition to do that. By the end of the summer, nothing had worked. And I took that as a sign that I was never going to be a scientist. I had liked science and math, but I had proven to myself by my own incompetence that I could not do an experiment to save my life. So I finished my college years, I took interesting courses, and then I had to get a job. I did a whole bunch of different things. One month I was a janitor. Another month I interviewed in hospitals, people. Another month my family, my parents used to make dresses, and I went to various stores selling dresses for a month. And then eventually I became a high school teacher. And I taught high school for 2 years. And while high school students get summer vacation, high school teachers do not. You have to find a job. And a friend of mine told me about someone at Yale Medical School who might hire me for the summer. And I went and interviewed with him, and that led to my first successful paper, and it gave me a lot of confidence that I could come up with ideas and actually do experiments, and they didn't have to fail every single time that I did it. And that I really needed that boost in confidence, and it was something I liked anyway. I liked thinking about the problems. I like and I still like the ideas aspect of it, even if I'm a little clumsy in the lab. Maybe a lot clumsy in the lab. I really like the ideas, and so it was very nice to go in, and that's when I went to graduate school.
There's a lady over there.
Brilliant presentation, thank you. You just showed the connections, that GFP make connections between science and art. So I want to know your perceptions about the bridges and the frontiers between science and art. The bridges and the frontiers, the barriers between science and art after being a scientist dealing with this.
So, I'm not sure there are always people that are very narrowly constrained, but I know a lot of people, in my lab for the last couple of years, just actually this last week, there's an artist who now lives in Boston who used to live in Genoa named Dimut Sreba. And Dimut, she's a wonderful artist. It's conceptual art. And she's taken on as her goal to do science-related art. So she sent out 2 art projects on the European Space Shuttle already. She has a whole series of pictures of scientists that she's done. She's done things connected to art and science, and this is what her career is now, which is just beginning to really flourish. She's got an opening in New York City in a couple of months, so she's very much in it. So she comes to my lab about every 6 months or so and tells me the latest things that she's been doing. So I don't see any barrier. I know that I'm on the Council of the American Society for Cell Biology, and they are always thinking about how we can showcase science and art together and what this means. The New York Academy of Sciences, and I'm sorry they stopped this, but for many years, the New York Academy of Science had this wonderful magazine that they put out every month, and it was filled with wonderful science articles, but no figures. Every figure in every article was a work of art that they put there. So although I'm sure there are people that do narrowly look at one thing or another, I find it's very surprising. I mean, I know a lot of people, myself included, that are very interested in music, and as well as interest in science. I think we're people. I think you're going to find in any group of people, you could say the same question, I think, of lawyers. Why isn't there more of a connection between law and science, law and art? Right? Because you're going to have some lawyers that are going to really enjoy various types of art, and you're going to get others that are not. I don't think that's a problem. But I think you're getting at a larger problem, and that is, how do we tell the world our excitement in science? How do we get them to do this? I don't know the exact numbers, but there's a story told that some survey went and asked students in New York City where they like to go. What museum do they like to go to? And the museum for kids, I think 12 was the age. I'm not entirely sure, but at 12, the age was 12 and under. There was only one museum to go to. It was the American Museum of Natural History. After 12, it was the Metropolitan Museum of Art. That somehow art was considered grown up, science was considered something for children, enrichment for children. It's complete nonsense. But I think we do have to sort of change people's perception of this and try to say we really need to show people why we're excited about science, why we do the work that we do, why we believe it's important, why it needs support. And I think that in part there has to be more outreach by scientists, that we really do have to set up ways in which we can communicate with the world about why we do what we do.
I have a last question.
Thank you very much for a very nice talk. And also thank you for making my PhD and postdoctoral work possible. Neither of them would have been possible without GFP, so thank you for that. So you talked about the importance of basic research, and I'm 100% with you on that one. But with the current funding situation, it's often hard especially for young scientists, to get money to do very basic research. And I'm sure you know this, but in the United States, there's a major shift towards bioterrorism-related research a few years ago. And that's, in my opinion, unfortunate. Is there anything that we can do to change this, to distribute the money or the research funds more evenly? Or is there anything that's already been done in that sense?
I think very definitely one of the things that people have to do is they have to participate more in the political process. I think that scientists, graduate students, postdocs, researchers, all have to make a commitment of inviting government officials to come and see their labs, to see what they're doing, to talk about why they're excited about what they do. I know in the United States, I'm part of it, something called the Coalition for the Life Sciences. It was set up over 25 years ago, first by the American Society for Cell Biology, and then it was added to by the Society for Neuroscience and the Genetics Society of America, and now Howard Hughes Medical Institute are all part of this. And one of the things they do is every month they have a different scientist from somewhere in the country come to Congress and talk about his or her work, to say why what they're doing is exciting and to explain it. So we've had all sorts of fascinating people, including people from industry, that have gone and said, look, industry needs basic research. Industry needs this because that's our pipeline as well. And so it's the involvement, I think, is there. We have to tell people, whether it's involvement at a national level or local level, or as we do at Columbia, we have once every month, we have a restaurant that we've reserved at 5 o'clock when no one's eating. And we have one Columbia professor go and talk about his or her work in science. And this is exceptionally exciting for the neighborhood and the area. We get lots of people that are not part of the university because they want to know about this, and then they spread the word. So I think there are outreach things to do, not just with children, but with many different age groups, to really highlight what we're doing. And I think I have the confidence that that's going to enlarge things and bring people in. But participation, I think, is a very crucial aspect.
Marty, just one last thing. You mentioned in your talk that you don't like being told what to do. Perhaps you ought to tell people how you disobeyed instructions at the Nobel Prize award ceremony.
Well, I didn't really disobey them. I would never do that. No, when my prize was announced in October of 2008, one of my high school friends, who already had gotten his Nobel Prize, Bob Horvitz at MIT, when we talked, almost instantaneously was saying congratulations, he said, "You're going to see Paul Nurse's movie." And I said, "Oh, okay." And he wouldn't tell me anything more. And then a month later, but before the Nobel ceremony, I saw Paul Nurse, who at the time was the head of Rockefeller University. And Paul Nurse said, "You're going to see my movie." And he was very proud of his movie. And what was his movie? Well, when you get your prize in the concert hall, each Nobel laureate gets up, goes to the king presents you with your medal and your proclamation, you shake hands with the king, you bow to the king, you bow to the electors on the stage, and then you face the audience and you bow to the audience, and you go back to your seat. And apparently, Paul Nurse, who got his prize the year before my friend Bob Horvitz got his prize, did all of that. But when he got back to his seat, he put the thing down and went, and that's the movie, that's the part of the movie they showed the laureates the next year of what not to do when you are getting your prize. It sort of removed a little of the dignity that was part of this. I don't know if it's on the nobelprize.org site.
No, we don't have it.
It's too bad. In any case, this was very much in my mind as I was sitting on the stage waiting to be called up to the king. And I was thinking about it, but I didn't want to do something like that. That was, one, it's copying. That was not appropriate. And two, they didn't show the movie. I think they had forgotten about it as well, so it was just not used anymore. I think they figured people had grown up since that time. And I did all of this stuff. I bowed to the king and after getting the award, and I bowed to the electors on the stage. And when I went to bow to the audience, I looked out, and there, right in front of me, in the third row, were my wife and daughter. And so I looked at them, and I blew them a kiss. And then I went and sat down. The next night, I was at the palace. In this thing, you go to the palace and have dinner with the king and queen and about 100 other guests. And I was seated in a very nice position. I was seated right next to Princess Madeline, who's very, very good-looking young woman. And on my left was a much older woman, Countess Alice. Very nice. And as I'm talking to Countess Alice says in a very gravelly voice, "Yesterday at the ceremony, one of the laureates," I can't do this, "blew a kiss at the audience." And I thought what that was going to mean. And I said, "Okay, yes, that was me." And I thought, they're showing my movie next year. And then she said, "I've been watching these things for 35 years. This is the first time I saw that. I liked it." And then the conversation went on from there. And then one thing that they do, which is a little bit strange, in case you ever find yourself in Sweden with the king and queen, is that after dinner, after they've had dessert, you don't sit and drink coffee. You stand up with your coffee cup and you walk around talking to different people. And every time Countess Alice came within eyeshot of me, she would look at me and go. So that's the story he wanted me to tell.
Marty, that's a perfect end. Thank you very much.
Okay.